Good afternoon and good morning and good evening, everybody. Welcome to our third webinar in the Cosmos ID webinar series. I'm excited to have Scott Jackson here from NIST, and he'll be talking about standards for microbiome and metagenomics. Just a quick brief on Scott Jackson. He's been with NIST since 2014. And prior to that, he was a principal investigator with the FDA for 11 years. He's focused his career on characterizing the global genomic diversity of enteric pathogens with applications for food safety, bioforensics, and public health. At NIST today, Scott is the leader of the complex microbial systems, which is in the Biosystems and Biomaterials division. And he's currently focused on efforts to improve the microbiome and metagenomic measurements by organizing interlab studies, developing reference materials and reference methods, as well as in vitro tools to allow us to help better understand the microbial community's resilience and evolution.
So take it away. Okay. I assume you can hear me. Hi, good morning, everyone. This is Scott. Thank you for that very kind introduction, Manoj. Happy Cinco de Mayo, everyone. Hope it's 5 o'clock somewhere. Anyway, yeah, so as Manoj says, I'm going to be talking about some of the ongoing work at NIST, standards for microbiome and metagenomics, but a lot of my talk is going to be revolving around poop, so you're going to be hearing a lot about that. So I just want to quickly give a background on what is NIST. A lot of people aren't familiar with NIST. So we're a non-regulatory federal agency in the Department of Commerce. We're the U.S. Metrology Institute. Most developed countries have a metrology institute, and in the U.S. it's NIST. Our headquarters is in Gaithersburg, Maryland, about 20 miles north of D.C., but also have a large, beautiful campus in Boulder, Colorado. About 5,000 scientists and engineers strong. We're the oldest federal technical institute dating back to 1901, and we have about $1 billion annual budget. And our mission is to promote U.S. innovation and industrial competitiveness by advancing measurement science standards and technology, which is why we are positioned in the Department of Commerce. I want to start just by letting everyone know that in September of this past year, we hosted a workshop that was titled Standards for Microbiome Measurements. It really focused on clinical applications of the human microbiome, namely diagnostics and therapeutics. About 150 people attended, and the reason I'm telling you this is because the entire workshop was video recorded. It's currently available on the NIST website, and you can go back and watch the entire workshop at your leisure, as fast or as slow as you want. There are a lot of great speakers from industry, academia, government. FDA was represented to talk about how they're thinking about regulating new clinical products that are coming out around the microbiome. And if you want to find the workshop, just go to Google and search for NIST Microbiome Workshop. You'll find it. And we also had one in 2016 that was also video recorded, so you can see then and now. Okay, so really everything I'm going to talk about from here out revolves around this measurement pipeline, which is a metagenomic measurement pipeline. So I think of metagenomics as the 21st century microscope, and that it allows us to see things that we couldn't see previously. You know, it was really the availability of rapid and affordable sequencing capabilities, so-called next-gen sequencing, that allowed us to begin to look at these complex samples that people had never really looked at the DNA level. So, as I say, there's bias in every step of the measurement process. So how you collect and store your sample, remember these are living and breathing samples. So if you collect something and you let it sit on the bench for a month before you measure it, presumably it's going to change, not presumably, it definitely will change. So the first step really after you collect your sample is to extract nucleic acids. I'll just say flat out, there are no DNA extraction methods that are 100% efficient across all cell types. You know, some cells are harder nuts to crack than others. And different DNA extraction methods, whether it's a mechanical bead beating, you know, chaotropic agents, lysis-based, or all of the above, they all have their individual strengths and weaknesses and will perform differently on any given sample type. So after you have your nucleic acid, you move into your library prep. Maybe you're doing 16S amplicon sequencing. If you are, you're going to do some variable region or some combination of variable regions. Maybe you're doing whole genome shotgun. And if you're doing whole genome shotgun, you think, well, that's a less biased approach because you're really looking at everything. And arguably that's true. But in a WGS library prep step, many times there is a PCR amplification step, and we know that that can introduce quantitative bias and that all fragments don't amplify with the same efficiency. So after you have your library that's suited for your particular DNA sequencing technology, of course, you put it on your sequencer, and there's a half a dozen sequencers out there that use different chemistries. And again, they all have their inherent strengths and weaknesses, whether it's homopolymers or error rate, these sorts of things. Move on to read processing, how you begin to filter, quality filter, et cetera. And then finally into your analysis and interpretation of your data. And, I'm not going to talk much more about this throughout the talk, but I just do want to point out that this step really is called taxonomic profiling, where you look at your reads and you map them to a reference database and you catalog what is in your sample. The software tools for doing that we call taxonomic profiling tools. There are over 100 that have been developed by academics, by industry, and they all use different algorithms. They use different statistical assumptions. Many of them use different reference databases. And the bottom line is that these 100 different tools will give you 100 different answers for the same raw data set. And just wanted to point out that each of these steps is a potential source of bias. And at the end of the day, where you have your bar chart or your pie chart that's describing the taxa that are present in your sample and their relative abundance, think about how representative that is to what you started with, considering the steps of bias, the methodological variables and the bias that they may introduce. So I want to start off by talking about an interlab study that we co-hosted a couple years ago. It was called the Mosaic Standards Challenge. It was launched in May of 2018. And the Mosaic Challenge is an international interlab study that's designed to assess the multitude of methodological variables that are associated with microbiome measurements and how these impact the results. And I used to show the slide saying we're currently recruiting new participants, but we are no longer, the challenge is closed. So it takes a village to pull off such a Herculean event. And I have to say this was really spearheaded by Janssen's Human Microbiome Institute, namely Dirk Gevers. If you watch our 2016 Standards for Microbiome workshop video, Dirk talked about establishing this interlab study. And sure enough, 2 years later, it was able to launch. So Janssen recruited the help of some necessary partners. So the Bio Collective is a stool repository, and they were able to make the material that I'm going to talk more about later that was used in this interlab study. They recruited the help of DNA Nexus, who built a custom cloud analytics platform that allowed the data for this particular interlab study to be housed, shared, analyzed, and visualized. We helped by helping design some of the study, and we made some DNA mixtures that were used as part of the materials that were shared as part of this interlab study. And DNA Genetech provided their proprietary storage buffer that the Bio Collective used to stabilize the material that was used. So what is the material? This is exactly what it looked like. If you participated in the study, you would have gotten an envelope that contained these 7 tubes in your lab. These are 5 poop samples that came from 5 different donors. So they're human poop samples from different humans. So they're arguably very different with respect to their taxonomic content. And these are the 2 DNA mixtures that NIST made, and they're purified genomic DNA from about 20 strains, and we mixed them at known relative abundances. So these serve as ground truth. And I want to point out that these poop samples, while we may measure them 100 times, 100 different ways and get 100 different answers, they will never be ground truth, and that we can't point to any one answer and say this one's right and everyone else is wrong. So poop will never be ground truth with respect to its taxonomic abundance. But we can make DNA mixtures that we are confident in that are ground truth. although obviously these don't challenge the DNA extraction step. So if you participated, what would you have done? So you would receive the samples in your lab, those 7 tubes that I showed. You process the sample using your standard methods. We didn't in any way want to dictate the methods or protocols that you use. Use your methods, or try multiple different new methods, of course. Sequence either 16S or WGS. Again, use your standard workflow. Then after you have your raw sequence data that comes off your sequencer, you upload that to a Mosaic biome analysis portal. And this, again, is the custom portal that was built by DNA Nexus specifically for this challenge. It did a real-time analysis of your data and displayed the results, and I'll show you a little bit of that later. And also importantly, you know, very carefully described sort of what the point of this was, and it was to understand the impact of methodological variables. So in order to really make sense of all of this, we need the metadata. We need every step of the process that you use, from how you did your DNA extraction to how you stored your samples to how many minutes you vortexed and what was your dry bath heat set to. These are the granular little nuanced details of the protocol that we wanted to capture. And after you submit your metadata and your raw data, you compare your results to those of your colleagues. This is a screenshot of the metadata reporting sheet. There were about 100 metadata values or metadata questions. I'm just looking at some of them here. One says, did you perform amplicon or whole genome? Obviously, that's important. Where samples were samples stored between receipt and processing? How are samples typically transported? How was the base DNA extraction protocol used? So these are the types of questions, and then across the different samples. And if you did process the different samples different ways, you could capture the differences in your methods. So once all of the data had been uploaded to the Mosaic portal, there's interactive tools that allow you to look at things like the metadata overviews. So here we're looking at all of the data that had been submitted from this challenge. It looks like about 175 had done Illumina MiSeq sequencing, and 60 or so did Illumina NextSeq. And you can use these drop-down menus to choose every possible methodological variable to see how it was used through this study. And then, of course, you can explore your data. So it uses the Emperor visual analysis tool. So this is a 3D PCA, and you can spin it. And again, there's drop-down menus that allow you to select the methodological variables, and it will color code the samples according to the methodological variable that you choose. So the challenge ended earlier this year, as I mentioned. It ran for just under 2 years. In total, we captured information from about 55 participants, which is large for an interlab study. A lot of interlab studies that we run at NIST have 7, 9, 11 labs, so 55 is relatively large. Those 55 labs generated slightly more than 700 FASTQ files, which is about a terabyte base, with the associated metadata, of course. About 2/3 of these labs did 16S amplicon sequencing, and the rest did WGS. Data analysis is underway for publication, but I'm going to show you a glance at these Mosaic challenge data in a very 50,000, 10,000 foot view. So first I want to show you data from the Mosaic biome portal. This was, of course, the portal that all of the data was originally submitted to. And again, I showed you this before. This is the 3D PCA plot. This doesn't look very informative. And I will say, most likely the reason is because it is showing all of the data that is the 16S, the WGS, and trying to show all of that in one plot. And of course, the 16S doesn't cluster well with the WGS, even if it's the same sample. So, we could play with this and filter out the 16S and just look at the WGS, and things start to clarify a little bit. But just an example of the screenshot of the data from the Mosaic portal. Let me mention, so the 3 glances, one from the Mosaic portal, the second from an in-house analysis that we did at NIST, and then we also gave the data to Cosmos ID, or I should say the Cosmos ID accessed the data, and they did their analysis for it on their platform. So Mosaic, Here is my in-house bar chart analysis. So I mentioned there were 5 poop samples and 2 DNA samples. This is shotgun data of poop sample 1, poop sample 1, poop sample 2, poop sample 3, 4, 5, and 2 DNA samples. And again, this is the WGS data. Here's the 16S data, obviously a lot noisier. And one reason why this might be noisier is because we didn't prescribe any particular variable region. So you might be looking at 16S data that was captured from different variable regions or multiple variable regions. Again, poop sample 1, poop sample 2, 3, 4, 5, and 2 DNA mixtures. Next, we use the Cosmos IDE tool, and I'm going to attempt to show you that. Okay. I assume you can see this, and if you can't, then Manoj, please jump in and let me know. So this is the WGS data from the Mosaic Challenge on the Mosaic portal, okay? And we can see that things are starting to cluster really nice. So here's poop sample 3, here's poop sample 1, poop sample 2, 4, 5 DNA samples, okay? So the DNA samples are relatively simple compared to the poop, okay? So there's only like less than 20 genomes in here, whereas of course, we know there's many species and strains in poop. So this is the beta diversity. If we look at Bray Curtis, things begin to look a little different, but again, they're clustered. Alpha diversity. So I thought this was interesting. Alpha diversity, of course, is a measure of taxonomic richness within a sample. And of course, the poop samples are much more diverse than the DNA samples. So here are poop samples, and then obviously the green moves into DNA, and then we're back up to poop, and so on. I thought that was interesting to see. So again, this is the 16S. I'm sorry, this is the WGS. And then over here, I'm clicking on the 16S data. And as I showed you in the bar plots that we generated at NIST, the 16S data is much noisier than the WGS data. And again, probably because different variable regions were used. But the DNA samples that were generated in NIST cluster much differently than the poop samples. What else can I show? So there are some bar plots. At any rate, we were just able, the Cosmos ID worked very hard getting this data up, and really I just saw it for the first time yesterday, but it's really a lot of fun to play around with. And we are going to use these data as part of our data summarization in our analysis and our paper. So let me go back to presentation mode. Okay, hopefully everyone sees that. So I just captured these screenshots in case for some reason I switched to the web browser and it didn't work, but this is essentially the same thing I just showed you. 16S, WGS, and 16S. Okay. So I used to use the slides to recruit people to join, but we're no longer recruiting. As I said, we ran for just under 2 years and collected data from about 55 labs. So I'm going to move on now to reference materials. So at NIST, we have a diverse suite of reference material in this little display cabinet in our lobby of our headquarters building. You can see some of the NIST reference materials. We've got standard reference cigarettes. We've got meat homogenate, which is also known as potted meat or spam maybe. We've got industrial sludge. We've got domestic sludge. We've got Portland cement, and peanut butter. Some of these might sound ridiculous, but they were all developed for very important and specific industrial applications. There's an industry out there that needs these and asks for these and uses these. So that's how NIST works. Being in the Department of Commerce, we see industry as our stakeholders, and we develop reference materials to help industry advance. We don't have mock communities at NIST, although we are working on some cell enumeration techniques and things like this. But I do want to point out that there are microbiome reference materials available outside of NIST from companies like ATCC. They've done a great job at developing a catalog, a suite of different mock communities that represent the skin microbiome. And then I think they've got the vaginal microbiome, and they've got a virome. And I don't have any financial stake in ATCC. I can't validate the quality. I don't verify the quality of these. I'm just pointing out that this is a resource available to you. Zymo is also a company that makes microbiome standards, specifically mock communities. And this is a screenshot from their website, and they've got, you know, a catalog of different mock communities, whether they're DNA or whole cells. And I'm pretty sure this screenshot is out of date, and they probably have even more today. So again, I don't have financial interest in Zymo. They don't pay me anything. I'm just pointing out that this is a resource available to the community. So one of the reference materials that we are working on that probably will be the most odd reference material that NIST has ever made is a human gut microbiome reference material that is human fecal material. So we're working on standardized poop. So a reference material needs to be homogeneous and stable. Think about fecal material and its biological complexity. And when I say stable, I don't mean stable for 6 months. I mean stable for 6 years. We don't have the capabilities to remanufacture new batches of stuff every 6 months. We have to make it once, we have to make it right, and it has to last for years. So fecal material is a very complex biological material made of small molecules, large molecules, and living microbes, viral particles. Think about like milk in your fridge. And what if I said, process your milk in a way that is stable for 6 years. It doesn't change for 6 years. That's a tall task. And that's how we're thinking about stabilized poop. So we are doing stability pilot studies. And I just want to point out some of the considerations in doing these. So manufactured material needs to be homogeneous and stable and amenable to the various measurement platforms. Poop, of course, as I just mentioned, it's inherently inhomogeneous, it's heterogeneous, and it's unstable. It's probably the worst case scenario for a reference material. And just to be clear, when I say homogeneous, I mean that the microbial taxa and the metabolites are the same across multiple aliquots. Stable means that metabolites and microbial taxa don't change over time. Okay, so think about volatile organic compounds that are part of the metabolome of fecal material. And the ideal preservation method will leave cells intact, and the integrity of the cell wall will not change over time. So, you know, you can imagine if you store the material for a few years and the integrity of the cell wall is being compromised slowly over those years, you know, your DNA extraction method might yield more DNA after 2 years than it did at the beginning. And you may think, oh, I'm getting better at my DNA extraction. But really, the material is just changing in a way that allows your extraction method to be more efficient. So these are the factors that we're considering and thinking about in making a human fecal reference material. So I'm going to describe the poop stability study that we did in 2019. It's quite simple. We took some poop, we mixed it with water, and we put it in a Ninja blender. And I want to say that we've partnered with the Bio Collective to do all of this, and they've been doing some custom work specifically for us. And they are really the industry experts at doing these sorts of things, that is processing and homogenizing fecal material. So now we've got our blender, Ninja blender full of homogenized poop, or we presume it's homogenized. We wanted to test different stability or storage methods. So we took some of it and we mixed it with the proprietary commercial stabilization buffer. This is the common one that's available. We just didn't want to put the brand name on it. We mixed some of the poop with this commercial stabilization buffer, and we made 4 1 mil aliquots of it. Okay, so these are 4 biological replicates of material that had been stabilized using this commercial buffer. There's another company that makes another commercial buffer. You can probably guess who they are. Took some of the same material, mixed it with this commercial buffer, made 4 biological replicates of material that had been stabilized using this buffer. Then we took some of the material, it just in water, we call it neat, stuck it at 4 degrees. Took some of the material, lyophilized it, and again, 4 biological replicates of each. And then we took some in water and stored at -80. So these are 5 different preservation methods that we are really looking to assess how they affect the measurement process. So those 20 tubes of DNA, 5 times 4 is 20, yes, that were divided among 5 different preservation methods. We stored that material in the fridge for 4 months, and after 4 months, we measured it using 4 different measurement platforms. We did NMR-based metabolomics on it. We did metagenomics, of course, via DNA extraction of the material. We did metabolomics using liquid chromatography mass spec and metabolomics using gas chromatography mass spec. So 4 different measurements on each of those 20 tubes. So I'm going to show you data from the 16S. I used to show bar plots, but I realize it's not that informative. So basically, we just extracted genomic DNA from each, did 16S amplicon sequencing, did a dotted 2 analysis on it, and then we looked at the data via 2D PicoA plots. And what we can see here, based on the coloring, are the different preservation methods. So the taxonomic profile clustered based on its preservation method, suggesting that that preservation method has an impact on obviously the taxonomic profile, but probably the impact on the cellular integrity. One method was much different than the rest, and this is the 4 degree. So if you take poop and mix water with it and stick it at 4 degrees, those live microbes in there that are still around food particles and fiber, they're going to continue to eat and process that stuff. So it's our hypothesis, I think it's probably a correct hypothesis, that at 4 degrees the material changed because the microbes were still alive and doing the things that microbes do, like eat food and metabolize and divide. We also, as I mentioned, did gas chromatography mass spec to look at its metabolomic profile. And what I want to show here is that within the 4 biological replicates within a preservation method, so minus 80 degrees, for example, or lyophilization, those 4 biological replicates within those were very good in terms of homogeneity. That is, the metabolic profile generated by GC mass spec was nearly identical, or I'm showing it here, so you be the judge. The metabolic profiles were overlapping, nearly indistinguishable for each of the biological replicates, suggesting that those 4 biological replicates were indeed homogeneous, right? But when we looked at a different preservation methods, and now we're comparing lyophilization versus minus 80 or any other method, we see that the metabolic profile changes. So just like we saw with the 4 degree with the taxonomic profile, when we look at the metabolomic profile using different storage methods, different preservation methods, we see different metabolic profiles, okay? So the preservation method matters. We're not saying one is more correct than the other, we're just simply pointing out that they're different. And here's the, again, PicoA plot of those GC mass spec profiles, and again, they're clustering based on their preservation method. Excuse me. Drink of water. I mentioned that we also did NMR-based metabolomics. This is some of the data from those 5 different preservation methods. Something interesting to note here is that the proprietary storage buffers that we use, they have high concentrations of surfactants and salts and things like this. And they really interfered with the NMR measurement in a way that our NMR guru, Sandra De Silva, was really unable to interpret because of the interference from these components of these proprietary storage buffers. But aside from that, really it was the same conclusion that we saw from the GC metabolomics, and that is there was good reproducibility within a storage method, but different storage methods gave different NMR metabolic profiles. And lastly, this is the LC data. So I'm doing it on time. I'm doing fine. This is the LC data, and the conclusions are the same story. Within a preservation method, the LC profile was reproducible, but across preservation methods, we saw considerable differences in the profiles. It's also worthy of noting that, again, those proprietary storage buffers that have proprietary components, but probably high concentrations of salts and surfactants and things like this, actually broke our instrument. So Paulina Piatowski, one of our LC mass spec gurus, she said she actually, after injecting it, she had to take her instrument apart and clean off the ionizer part because the material had gunked it up, and it was specifically whatever material is in that proprietary storage buffer. So these proprietary buffers should be avoided for metabolomic analyses. And again, with regard to the proprietary storage buffers, I'm not saying whether or not they do a good job. They probably do just fine at stabilizing the material. It's just, we're not saying that they're not good at stabilizing. We're saying that the chemical components make them less amenable to these types of measurements. So what are our conclusions? You're like a broken record. I'll say it again. Within the preservation methods, the 4 biological replicates appear sufficiently homogeneous. So nod of the head to the Bio Collective. Their manufacturing process appears to produce sufficiently homogeneous material. The choice of preservation method impacts the downstream measurements. Each preservation method produced its own unique metabolic and taxonomic profile. Blah, blah, blah. The proprietary storage buffers, they're problematic. And, from a taxonomic point of view, we know that they can lyse some of the cells, and, lysed cells probably are stored, stable just fine, but we want to keep cells intact as much as possible. And lastly, if you're following closely, once we prepared the material, we stuck it in we stored it at its described storage conditions, minus 84 degrees, for 4 months. But there was never a T = 0 measurement. We didn't measure the material as soon as we aliquoted it. We just aliquoted it second fridge. So this wasn't a true stability study. And really, going back to the intentions, was to understand the impact of these stability preservation techniques on the downstream measurements. So that leads us to 2020 Poop Pilot Stability Study 2.0. And in this one, we went a little bit larger in scale. So in this case, we recruited 2 cohorts of donors. Cohort 1 was omnivores. Cohort 2 were vegans. Multiple donors per cohort. Why did we choose omnivores and vegans? Really because we just wanted 2 types of poop that we knew would be different with respect to their taxonomic profile and their metabolic profile. And while diet is known to have a very large impact on the contents of your gut microbiome, we felt that by choosing omnivores versus vegans, we would see the largest differences between these types of poop. Okay, so just like before, cleaned out the old Ninja blender and collected samples, homogenized them in the Ninja blender, and then we made about 500 aliquots of each cohort. Okay, so about 1,000 samples. And it was our intention to measure the stability for 12 to 24 months. But then something happened. It was called the coronavirus pandemic. So quite literally, the week that we made these aliquots, we were the following week sent home because of the pandemic. So we have these 1,000 aliquots. They're sitting in a minus 80 freezer in Colorado. And we missed our T = 0 time point. The point of this was to measure stability, from week to week, month to month, for out to 2 years. But it required us to take a T = 0 time point, and we missed it because we were sent home because of the pandemic. So now we've got 1,000 tubes of poop sitting in the fridge, and we're scratching our head asking what to do with it. So we've all been doing our best to respond to COVID-19, and with respect to fecal material, you've probably read that SARS-CoV-2 virus particles are shed into the feces of infected patients. And this creates some unique opportunities and challenges. And I'm going to quickly outline a few of those. So some unique opportunities and challenges. One, feces could be a new target for diagnostic testing, right? So currently there are over 140 commercially available COVID-19 diagnostic tests. They were initially validated on nasal swabs, but you can imagine that these same tests can potentially be revalidated in fecal material. And if this is the case, then we believe a fecal reference material is needed in order to assess the performance of these tests in this challenging sample type. So, got poop. Another unique opportunity or challenge, fecal microbiome transplantation. So every year, thousands of patients receive FMTs to treat infections like C. diff, of course, but they're also being used in clinical trials to treat a plethora of other health-related issues and diseases. To ensure future safety of fecal transplantation, donor material must be screened for known pathogens, and this is FDA enforced. There's a list of defined pathogens that have to be screened for, like HIV and hepatitis and these sorts of things. And last month or 2 months ago, the FDA released a safety alert regarding the use of FMTs in COVID-19 that includes, quote, testing donors and or donor stool for SARS-CoV-2 as feasible. So now FMTs will need to be screened for SARS-CoV-2 viral material, and this will require assay validation that requires fecal reference material. And last, unique opportunities and challenges with respect to COVID-19, and that is monitoring sewage and wastewater treatment plants. I'm sure many of you have heard this. It's nothing new. There's papers dating back to the 1980s with them doing this with viruses like HIV and others. But local public health labs can monitor sewage treatment facilities to measure the rise and fall of COVID-19 in the community. So community-wide assessment of viral load, so to speak. Papers have been published on this. One was recently quoted as saying, "This visibility is also going to help us predict a second wave of outbreaks." And in doing this, one would imagine that it would be nice to have fecal reference materials available to validate assays and to understand reproducibility across laboratories, impacts of methodological variables, so on and so on. So what are we, going back to the 1,000 tubes of poop that we have sitting in our fridge that we were going to use for a time course study, but we now are looking for a new use. We want to engage the stakeholder community, that is you all, to see how we can most effectively use this material in response to COVID-19. So we are hosting a virtual workshop. The running title is Standardized Fecal Material for Validating SARS-CoV-2 Fecal Testing Methods, Use Case Scenarios. We want to hear from the community how this uncertified material that we have, it's not a NIST reference material, it's not a standard reference material, it's just some material that we have available that we think we could share with the community to assist in the response of COVID-19. But the point of the workshop is not for us to tell you what we have or what to do. It's to hear from you to tell us how best we could use this material. Maybe we host an interlab study of methods that have been developed for nasal swabs, and we get a half a dozen of those, and we look at their analytical performance at testing SARS-CoV-2 in these fecal material. I should point out that the cohorts that we collected from were from 2019. Presumably, they're all free of coronavirus because the pandemic hadn't reached us to that point. So they're not positive controls, but they could be used as negative controls and, more importantly, as spike-ins. So there are coronavirus reference materials that are available, again, not yet from NIST, but ATCC, and there's like a dozen companies around the world that have been developing SARS-CoV-2 reference materials. You could buy those, you can spike them into the poop at known quantities, so they're quantitative reference materials. You can understand limits of detection, sensitivity, specificity using these materials in a standardized way, in that everybody's testing the same material. So if two labs get different results, it's not due to differences in the matrices that they're using. So join us on June 16th, 1 p.m. to 3 p.m. We're inviting subject matter experts to speak about those three opportunities: sewage wastewater treatment, FMTs, and applying existing diagnostic methods to fecal material. Okay, so look for an announcement from us in the future on that. I'm wrapping up here. I just want to point out that we're always hiring, in that we have a postdoc program that's run through the NRC. It is a very competitive program, but it is also very prestigious, and our postdocs go on to do great things like become the director of NIST and win Nobel Prizes. We are looking for postdocs that want to work on standards for microbiome and metabolomics, a lot of the stuff you've heard me describe. Also, we have an important program on pathogen detection, and it's all sort of the same thing. It's detecting microbes in complex matrices. As I mentioned, it's competitive, but it's worth the effort. It has a relatively high starting salary for postdocs, $72,000. I think the NIH standard right now is about $55K. And if you're interested in working with us and submitting an application, which is a research proposal, email us at nistmicrobiome.nist.gov. This is the wonderful and talented team that I've had the pleasure of working with. We call ourselves the NIST Poop team, unaffectionately, the NIST microbiome team. And most of these are NIST people, but I do want to point out that these are the biocollective people who have been so important in everything that we've done, because we can really, work with them in a way to make manufacturing material in a very specific, prescribed way that suits us, in that it allows us to understand impacts of different manufacturing processes and how they impact the stability and homogeneity. And of course, we've worked with Cosmos ID for years. The great colleagues, Nir Hassan has unfortunately left Cosmos ID recently to start his own company, and that's great. Pat him on the back and support him. But Manoj and his team have been great, and that's all I have. With that, I'll take questions.
No one's yelled in the chat yet. All right. Start. All right. Questions on the school stuff. Questions on the school stuff. When comparing methods, can you just compare with initial results versus long time to determine which methods help the least change? From James.
Manoj, your connection is really bad. You're cutting in and out.
Okay, I have a.
Maybe I should read the questions myself. Yeah. Oh, you're calling me. Hey. Okay. Why do we think the lyophilized are more homogeneous? I don't think I made that conclusion. I don't think I made that statement. If I did, I didn't intend to. All of the material was homogenized in a Ninja blender, so presumably it was all homogenized the same way. So I wouldn't expect the lyophilized material to be any more homogeneous than the rest. Yeah. Yeah, give me just a second. I'm going to put my earbuds in. All right, now we're hands-free. Can you repeat that? No. So for Poop Pilot Stability Study 1, which we did last year, we didn't take a T=0 time point. Intentionally or unintentionally, it was really just to assess the impact of the different preservation methods and how they impact the measurements. But for stability study 2, which we were starting this year. It was perfectly our intention to characterize the material before and after. And like I said, we weren't able to do that. Sure. I mean, they're DNA-based viral particles, so they'll presumably be seen in the extraction and the metagenomic profile. Okay. Someone asked if phage could be seen in the taxonomic profile of the fecal material. I believe that was the question. And DNA, most phage are DNA-based, and yeah, that DNA should be extracted just like any other DNA, and we should see it in the taxonomic profile. Yes, we will be publishing these results at some point. And can you repeat the second? Oh, sorry, I have to repeat the question. Can you repeat the question, Manish? Sorry. Okay, so the question is, what storage method was most similar to -80? And I'm presuming that the basis of that question is that -80 is sort of the gold standard for storage. So what was most close to that? Honestly, I can go back and look at our looks like lyophilized. Yeah, lyophilization was most close to -80. So it requires an RNA. Oops. So the question was about RNA viruses and how well they show up in shotgun sequencing. To see RNA in metagenomics, you have to do, first of all, an RNA extraction, which is slightly different in that you have to be a little more gentle with material. And then you have to do a reverse transcription step to make it cDNA. And once it's cDNA, then you can sequence it in a traditional way. And we do know that reverse transcriptions in general are relatively they're not that efficient and they're error prone. So there's some problems that might be introduced from a reverse transcription step. But other than that, I will say that the Oxford nanopore technology does allow direct RNA sequencing, although I don't know if anyone's done metagenomic sequencing using Oxford nanopore. But yes, we can certainly see viral RNA particles using metagenomics if we incorporate a reverse transcription step. Yes, that is. So the question is, I keep forgetting. The question is, since the new poop samples have been sitting for months, are you worried that they don't represent what they originally started out to be? And the answer is absolutely yes. That is our worry. That is why we've decided not to continue doing our stability study, because we didn't capture that t=0 time point. And I want to back up and answer a previous question because I think I answered it incorrectly. Someone had previously asked, do you measure what the material looks like before and after preservation? And I think there's two parts to that question. One is the raw feces that comes out of the humans. Do you look at that as the before, and then you process it and homogenize it and you stabilize it, and then you measure it after that, which may be a t=0 in and of itself, and that it starts the preservation method. But we didn't account for that in either of our stability studies, but it is something we've talked about and recognized that's probably needed, because it would be interesting to see how raw stool measured taxonomically differs from homogenized, stabilized stool. So the question is about specifically stabilization in metabolites and metabolic profiling. I should mention that in September, when we had our workshop, we also had a smaller one-day workshop that focused specifically on metabolomic measurements of fecal material. And we invited in, a couple dozen subject matter experts from around the world who have been doing fecal metabolomics to really get a data dump, a download from their brain on their experiences in doing this. And one of the things we learned from these experts in their experiences, there's no way to store fecal material that will preserve 100% of all molecules, of all metabolites. From what we were told, something's going to give. These small volatile organic compounds, you put them at -80, you put them at -400, like something's going to change with respect to the metabolites. So when we say that the material, first of all, we're not saying it's stable, but that's the whole point of us doing these stability studies, is to understand what stability looks like. But I suspect stability is going to come with some asterisks in saying these things appear to not be stable, and possibly these things are stable. But we're learning as we go, but we certainly expect that some metabolic compounds will probably not be entirely stable no matter how we store the material. Yeah, so Manoj is asking if we're going to continually test the material. Yes, and that was the point of those 500,000 samples, Manoj, is that we were going to measure a few every month. We were going to measure them metabolomically and metagenomically every month and sort of see a time course. And that way, if something's changing, you sort of see a drift, Yeah. So the question is, when we do make our fecal reference material, is it going to be from an individual donor, or will it be from a cohort of donors? We are imagining it will be from cohorts, and it will probably be a suite of material that represents different cohorts, like, for example, omnivores and vegans. And maybe we select some different cohort types because we want different types of poop. Most definitely it will come from cohorts of people, so no single individual. And the second part of the question was, does NIST have a reference human, I think was the question, and the answer is no. So like, for example, we have human genomic DNA reference material, but it came from one of the Personal Genome Project donors that's part of the PGP from Harvard. It's one of these people who completely consented their DNA, and that's the one we use for the NIST human genome reference material. And in fact, we have a suite of human genomic reference materials now, and they all came from the PGP. So. The question is, I guess, what have we learned about microbiome measurements that drug developers, pharma companies can use to fast track clinical applications, I presume. So microbiome measurements with metagenomic measurements are not accurate, but they're precise. And, if you remember the analogy that we've all seen of the dartboard with the darts going in it, and it shows the difference between precision and accuracy. Accuracy means that you're reproducible, but might not be on the bullseye, but at least you're reproducible. Accurate means you're on the bullseye, but you might not be very reproducible in that, you're sort of all over the board. With respect to microbiome measurements, when we measure them in an individual basis, that taxonomic profile is not accurate. It's not correct. To quote a colleague, Ben Callahan, all microbiome measurements are wrong. But they're reproducible in that if you use the same lockdown method and measure across a set of samples, the relative differences between those are most likely real. If I see a twofold change in firmicutes between me and Manoj, and I use the same method, the same methodological pathway, those changes are likely real. Now, the absolute levels of firmicutes in Manoj and I that are measured are probably not accurate. So that's why people don't try to interpret a single microbiome. It's always relative to something. We're looking at cohorts, or we're looking at differences between individuals. So that's an important consideration. And if we are to move into microbiome diagnostics in a traditional sense, in those cases where you are diagnosing someone or measuring a microbiome as a clinical diagnostic, it has to be accurate because it has to be able to be harmonized across the world. A lab in Europe that measures a microbiome as a diagnostic can't get a different answer than a lab in California. If you think about traditional diagnostics like cholesterol or creatinine, you measure a person's creatinine in Germany, you're going to get the same answer as you get in the United States. And that has to be the case for microbiome. So, yeah. How far out is that? There are groups that have made progress in a very innovative and way. And I'll point out that, for better or for worse, 23andMe had a clinical diagnostic. It was prescribed by your physician, and they would measure your sample, and then they would give the results to your physician, and you could go consult with your physician. But if you look closely at the fine print, it wasn't a true microbiome profile. It wasn't a complete taxonomic profile. But what they had done is looked about 20 organisms that had been identified through previous research as being important or associated with something. And they validated individually each of those 20 organisms. That is, they looked at their sensitivity, their specificity, the performance of their measurement on how accurate they were performing on these 20 organisms. So those were accurate microbiome measurements for those 20 organisms. Now, how do we begin to expand that list from 20 to 20,000? That's part of the challenge. The question is, what are the advantages of using stool over nasal swabs? And I'll say maybe not much, because the literature that I have read shows that not all infected people are shedding virus in their fecal material. And I've read some reports as, under 50% of people infected with coronavirus actually shed it in their feces. So indeed, it's probably an unreliable diagnostic. Although there may be situations where it may be more useful. Don't have a good answer for that. It may not be, it probably isn't a reliable target for clinical diagnostic, but I'm sure someone out there is working in a space where they're imagining that it could be useful. The holy grail, I believe, is our mock communities that truly represent a real microbiome that you find in the world, whether it's feces or whether it's soil. These things have tens or hundreds of different species and, unfathomable number of strains within a species. To be able to engineer from the bottom up communities that truly represent a microbiome, that are ground truth, that's, I believe, the holy grail. The mock communities that are out there from ATCC and Zymo, they typically have 10, 20 organisms in it. And, that's a challenge to, make them that complex. So what we are working on at NIST is we put a lot of time and effort into cell enumeration methods and understanding the accuracy of cell enumeration methods. Bacteria can be very difficult, if not impossible, to count as individual small microorganisms, because not all microbes behave like E. coli. They don't form these nice individual countable units. A lot of bacteria form chains and clumps and filaments and do these very weird things that make them nearly impossible to count. So mock community development is 100% reliant on our ability to accurately count bacteria, and not all bacteria are easy to count, and some are damn near impossible. Give me the Cliff Note version of that question, Manoj. Okay, how do we work together in an international way to move standardization forward? Very importantly, obviously, NIST isn't the only one working on this problem. There are metrology institutes around the world, like NIST and other countries, that are working on microbiome and metagenomic standards. We work closely with our Japanese colleagues at the Metrology Institute in Japan, who are kicking off big nationwide surveys of the Japanese microbiome, and they've developed standards. I think it's going to take time, of course. But I do see more international coordination, and that, in our workshop that we had, I believe there were about 20 international participants. So I guess I don't have a good answer for that question, but I feel like we're moving in the right direction. We do have a long way to go, though. When I was just saying, we're out of time. Thank you for joining us. No, thank you for having me. Go ahead, Manoj. I'll translate for you. Yeah, thanks to the audience for questions. We'll follow up. I guess you have a log of all these questions, and we can get to some of them. Yeah. Okay, thanks everyone. We're signing off. Bye-bye.